Thank you for standing by and welcome to the Imugene Investor Webinar, following the announcement earlier this week of new data from the company's Phase 1b trial of its Azer-cel technology. Joining us from Imugene today, we have the CEO and Managing Director, Leslie Chong, joined by Chief Medical Officer, Dr. Paul Woodard. All participants are in a listen-only mode. There'll be an initial slide presentation lasting for approximately 15 minutes, and we'll take questions thereafter. If you'd like to submit a live question, please do so using the Q&A panel within Zoom. We've already received quite a few questions on email in advance, and we'll get through as many as possible. To kick off, I'll hand it over to Leslie.
Thank you so much, Matt. Good morning, good evening, wherever you're calling in from. Great to have you. We are excited to talk about Azer-cel this morning. So Imugene is listed on the ASX as IMU as a ticker. We are leaders in innovations in cancer treatments, and I can share with you why. So we have clinical stage cancer products in CAR T-cell therapy as well as oncolytic viruses. And we're currently at market cap around 500 million with a cash position of 93.1. This is currently without our R&D rebate, so that's soon to be here. We have 4 unique platforms, but we're going to really focus on the allo CAR T today, long life patent portfolio with disease areas now to include predominantly blood cancers as well as other solid tumors. So I'm going to hand it over to my incredibly capable and incredibly intelligent Chief Medical Officer, Dr. Paul Woodard.
Thank you, Leslie. So today we'd like to walk you through some of our recent data since Imugene acquired Azer-cel. So when we acquired Azer-cel in late last year, we've enrolled 10 patients to date, actually now 11. 10 of these patients were treated with Azer-cel. These were patients with diffuse large B-cell lymphoma who had failed autologous CAR T relapse therapy. And these patients were treated in 2 different cohorts. So the first cohort was using lymphodepletion, which is chemotherapy before Azer-cel is administered, which was standard dose fludarabine and a slightly higher dose of cyclophosphamide. We then transitioned to cohort B, in which we had the same lymphodepletion chemotherapy, the same dose of Azer-cel, and added a low dose of interleukin-2 for 2 weeks. And I'd like to go over some of the data that we've seen with this. So if we combine the 9 evaluable patients who've had at least 28 days of follow-up, we've observed a 44% response rate and a 33% complete response rate. If we focus on cohort A, which was with no IL-2, we saw a response rate of 33% and a complete response rate of 17%. However, the durability of these responses was less than 60 days or less than 2 months. When we focused and opened cohort B to include low-dose IL-2 for 2 weeks, we immediately noticed a much better expansion rate of the CAR T-cell, which I'll show in just a moment. But more importantly, we noticed that the first 2 patients both achieved a complete response. And these are patients who have had 4 to 6 prior types of chemotherapy, including an autologous CAR T treatment, which they have failed from. And even more striking is that these responses are already more durable than they were without IL-2. We have 1 patient who's over 120 days in complete remission and 1 patient at more than 90 days. This trial is being conducted at 15 leading centers in the US, including Columbia, the University of Minnesota, Emory, and Moffitt Cancer Center. And we hope to have up to 5 sites open in Australia as early as in the next 1 to 2 months. Next slide. So Azer-cel has a manageable safety profile. When we give CAR T cells, there's sort of 3 things that we worry about. And since this is an allogeneic CAR T from another patient or another donor, we also worry about something called graft versus host disease, in which the T cells from the donor can attack the patient. We've seen no evidence of graft versus host disease in any patient treated with Azer-cel, and we've seen no grade 3 or higher cytokine release syndrome, or CRS. I'll walk you through these cohorts. So for CRS, which is a fever, can evolve to more serious things like problems with your blood pressure and other issues. We've only seen grade 1 or 2 CRS, which is manageable. With Azer-cel, we saw a 33% incidence of CRS, and with the addition of IL-2, it's a bit higher at 66%. The second complication that we worry about is neurotoxicity, which is called ICANS, which has a very long name, but it's basically neurologic adverse events caused by the CAR. With Azer-cel alone, we saw a rate of grade 3 or higher ICANS of around 33%, and we've not yet observed any ICANS in patients treated with the addition of IL-2 with cohort B. The other thing that we worry about in these patients, because these are mostly older patients who've had many types of chemotherapy treatments, are severe infections. And we've mostly seen only low-grade or grade 1 to 2 infections of 50 to 60%. We did see a 33% incidence in the Azer-cel alone cohort of grade 3 or higher infections. In the table, these sorts of adverse events, CRS, occur relatively early on, and they are short in duration, so between a day or a day and a half in duration. And then for ICANS, this also tends to occur early and has a relatively short duration, at least for the patients treated with Azer-cel alone. Next slide. So one of the reasons we think this is working better with IL-2 is IL-2 is a growth factor for T cells. It helps T cells survive and proliferate. And so if we look at cohort A, which is the cohort of dose level 4 of Azer-cel with augmented cyclophosphamide, but no IL-2, and here we're combining both 6 patients treated by Precision Biosciences and 6 patients treated by Imugene using the same dose level and the same lymphodepletion, but slightly different manufacturing processes. What we see is very little expansion, which peaks at day 7. And these CAR T cells are largely gone by day 10 to 14. With the addition of Azer-cel, we really see remarkably better expansion. This is more than 2 logs higher with the addition of low doses of IL-2 for 2 weeks after Azer-cel is given. And we see better persistence, better maximum concentration, better area under the curve. And at the longest point that we've looked to date, we have seen detectable Azer-cel now out to 60 days. This is likely correlating with the improved responses we're observing. Next slide. I guess one thing I should mention is IL-2 doing anything to the response? IL-2 has been studied in many types of cancer and has really only shown responses in patients with melanoma and renal cell carcinoma. So we don't think IL-2 is actually generating the responses, but it's helping Azer-cel grow better and last longer. All right, so this is a PET scan or positron emission tomography. It's looking at the metabolic activity or the glucose use of tumors. And this is a 60-year-old woman who was diagnosed with diffuse large B-cell lymphoma, stage 4, in April of 2012, and she has been treated at the University of Minnesota. And this patient has actually had 5 prior lines of therapy. That's a lot of therapy. They received R-CHOP, which is the standard of care for the first type of treatment for DLBCL. Then they received intensive chemotherapy, followed by an autologous stem cell transplant. Then they received additional investigational therapy, followed by their first autologous CAR T, which is Yescarta, after fludarabine and cyclophosphamide, and then one additional line of treatment after that. They had relapsed with all of these therapies. They came on our study and received Azer-cel after the augmented Cy conditioning and received low-dose subcutaneous IL-2. This patient did really well. They had no CRS and no ICANS. And you can see at baseline what we're focusing on. If you look in sort of the neck region of this patient, you see a lot of white lesions, which is where the tumor is. And you're seeing a lot of uptake of this tracer in that area. At day 28, we saw a partial response in which most of the tumor was gone, which converted to a complete response by day 60, and that's now been maintained to day 90. So this patient has done well with Azer-cel, with IL-2. Next slide. This is our clinical development strategy for Azer-cel. So Precision Biosciences initially did a phase 1 study with Azer-cel. They enrolled 84 patients from 2020 to 2023. In Imugene's hands, we have now enrolled 11 patients in the phase 1B portion with cohort A and cohort B. We intend to enroll 10 to 20 patients using the cohort B approach with IL-2, and then seek FDA clearance for a phase 2 registrational trial later in 2025, and then institute a randomized phase 2/3 registrational trial as early as the end of 2025 to early 2026. Next slide. I'll hand it back over to Leslie.
Thank you, Paul. So within the hands of Imugene wonderful clinical development team, we have really progressed all our therapies. You see a thread going across there, Genentech, which I think is one of the most innovative clinical development in oncology, and Exelixis, where I met Paul roughly about 18, 19 years ago. We worked together there when his daughter was 1 year old, and now she's going off to college, which is amazing. And, you know, within the second half of 2024, you can anticipate hopefully further news on Azer-cel, building on what the runway that we've already created. Our onCARlytics highly anticipated first patient in for the IT combo with Blincyto, and Vaccinia. So Vaccinia will have a second indication that we're going to expand into, and hopefully early and preliminary results from our BioTract expansion. And then 2025 and beyond, you can see it's littered with huge milestones, such as Azer-cel meeting with the FDA for that registrational clearance of phase 2/3 study, our onCARlytics getting that optimal biological dose and expanding into a phase 2, and then combining with a new iterative Azer-cel, possibly in the future, and then Vaccinia just across having that optimal biological dose running into a phase 2. So all those are quite near term, just around the corner in 2025 and then beyond. And I just want to reiterate the investment highlights for Imugene in that we have novel platforms, 3 in particular that are in the clinic running towards a registrational study or phase 2s, strong cash position, clinical data readout, significant readouts in the next 12 months, really, highly experienced and well-rounded team and development. And then we have a robust and broad patent portfolio. Thank you for listening to our presentation.
Thanks, Leslie, and to you also, Paul, excuse me. As mentioned, we'll now move on to questions from what's been submitted live and on email. So without further ado, I'll jump into those. Can you give us a ballpark estimate of the number of additional patients required to complete the phase 1B? For example, is it approx 5, 10, 20 or further?
We'd like to enroll 10 to 20 patients in cohort B. We've treated 5 patients to date, and we have others currently being evaluated for the study. I would say the other important part of that is not just enrollment, but you need to follow the patients for a minimum of 6 months, because we think complete response rate at 6 months is really the important piece. We'd like to see anywhere from a 30 to 50% CR rate that's durable for a minimum of 6 months.
Thank you, Paul. Do you have a revised estimated start date for a potential registrational trial? Previously indicated Q1 CY25. Is this likely to be sometime in 2026 now?
No, our goal is 2025. It may scoot over a couple of quarters, but we're still aiming for 2025.
Thank you. Was the previous 83% overall response rate in DLBCL trials achieved using the same dose rate that is being administered in the current trial? If not, can you give us an overview of how the current dose level came to be selected?
So the 84 patients, and of those, 18 patients were autologous CAR T failures. That was in various different dosing and manufacturing process. The one we're using has been pretty consistent from the last 6 dose that Precision dosed, and then we changed the manufacturing process slightly and received FDA approval back in November of 2023, and that's the dose we're currently using.
Thank you. Precision Bio had previously received orphan drug and fast track designations from the FDA for various indications. Do these still stand? And if so, do either of them cover DLBCL? Do you anticipate seeking any further designations for DLBCL?
We will definitely seek other designation from the FDA to start engaging with them. Those other designation from the FDA was gotten for other indications like leukemia, not necessarily a DLBCL.
Thank you. Their announcement talks about patients having failed prior treatments, including autologous CAR T therapies. Is this relapse setting autologous anti-CD19 CAR Ts or other? Can you elaborate there, please?
These are right. Autologous CD19 CAR failures.
Thank you. Have all patients for the phase 1B been recruited, and have they all been dosed now? If not, when can we expect these activities to be completed? I think this doubles up slightly on earlier, Paul.
Yeah, that's the same answer as before. So we need to enroll a minimum of 10 to 20 patients and follow them for a minimum of 6 months. And we've treated 5, and we have others currently being evaluated.
Thank you. And related is, how long will it take Imugene to analyse phase 1B trial data, prepare an FDA submission paper and submit it for review?
Well, we have been looking at data as it comes across. As Dr. Woodard stated, we want at least 6 months of secure CRs before we run up to the FDA. So, but we plan on engaging the FDA a bit earlier than when we amass the data, so that we can start engaging in conversations with them.
Thank you. And again, related to that, how long does the FDA take to review the material, meet with Imugene, and then provide a decision on a phase 2/3 registrational trial?
It's about a 70-day process from the time you apply for an end of phase meeting to actually having the meeting with the FDA.
Thanks, Paul. Regarding the result for the 2 patients to date, how likely is it that their recovery is down to factors other than the treatment received as part of the trial?
Extremely unlikely. So these are people who failed autologous stem cell transplant in some cases, autologous CAR T therapy, multiple lines of chemotherapy, It would be miraculous to see a response without any sort of treatment.
Thank you. The next question is, what is the approximate market worth for the treatment of this form of cancer?
About 30,000 patients are diagnosed in the US alone with non-Hodgkin's lymphoma. 18,000 of those are likely DLBCL, and then they go through several treatments. We know that auto CAR T fails at roughly about 60 to 70% of the time. And so if we were to address just all, including all the addressable market just in the US alone, we're looking at a modest amount of $2.5 billion.
Thank you. How does Azer-cel's safety and efficacy profile compare to other allogeneic CAR T therapies?
We think the safety compares very favorably with other CAR Ts, not just other allogeneic CAR Ts.
Following on from that, what makes you think Azer-cel might be the first to get FDA approval?
Because we have a recommended phase 2 dose, we have a final manufacturing process, and we think we have a pretty direct path to meeting with the FDA in a registrational trial.
In cohort B, are you using a low dose, i.e. via under the skin injection, or a high dose administered intravenously over a number of days?
I think this is about IL-2. So the IL-2 is given as a low dose. It's 1 million units subcutaneously daily for 14 days. So it's a low dose for 2 weeks.
And it's a fraction of the recommended dose, yes.
It's much lower dose than's used in other cancers.
Thank you. Another question, there was a mention of another patient that was about to be evaluated within this trial. Has this subsequently happened?
Not yet. They're still within their days, yeah.
Thank you. Another question, can you explain the durability data in the Azer-cel results, i.e. what is durability?
That means you have a PET scan 28 days after the CAR, and then you re-scan these patients periodically. So in the beginning, it's frequent. So it's at month 2, at month 3, and so forth. And so you still see a complete response over multiple scans, multiple months apart. That's what we're talking about. You can't just have a response that is there 1 month and then the patient relapses. You need one that the patients are in remission for a long period of time.
I mean, plainly put, durability is just measuring how long you are without tumor.
Right.
Thank you. Another question is, are you finding it easy to enroll patients? Are oncologists coming to you to seek out your trials?
I would say we are. So we have 15 sites open, and we'll soon have sites open in Australia. We're always looking at potentially expanding where we take the trial. And so we've actually enrolled pretty well, I think, overall for this study. And you have to remember, there was a period of time where we amended the study to add IL-2, and we really didn't miss time due to that. So I think it's enrolled pretty well so far.
Thank you. I think this question points to potential revenue, but the question simply is, can you charge patients during a registrational trial?
No.
Thank you. Another question for you, probably, Leslie. Is there any link between Imugene and the melanoma research being conducted in Western Australia?
No. We don't have sites in Western Australia. Vaccinia would be the only one that would be enrolling with melanoma patients. So I think not at this time, but I'll take a look.
Thank you. Another question is, do you still plan to trial oncolytics combined with Azer-cel?
Yes.
Thank you. Paul and Leslie, thanks for your time today. Leslie, I'll just hand it back to you to provide a closing comment.
Thank you, everyone, for joining the call. I'm quite excited about the announcement and the data to date. I look forward to following up with more data and more outcomes.
Thanks, Leslie. Thanks, Paul, and also to everyone for joining today. We'll update you with
Thank you.
more soon. Thanks.